Hello students, welcome to Read Med Prep Academy channel. Today in molecular genetics we are going to discuss about translation. What is translation? Translation refers to the process of polymerization of amino acids to form a polypeptide chain. The decoding process is carried out by the ribosomes that bind the messenger RNA and the charge transfer RNA molecules. Here in this image you can see the two subunits of the ribosome with the messenger RNA and the charge transfer RNA bringing the amino acids and the amino acids forming the growing polypeptide chain. At the ribosome, the RNA's message is translated into a specific protein. The messenger RNA is translated starting at the 5' end. After binding to the messenger RNA, the ribosomes move along it, adding new amino acids to the growing polypeptide chain each time it reaches a codon. Each codon is read by an anticodon on the corresponding transfer RNA. Hence, the order and sequence of amino acids are defined by the sequence of the bases in the messenger RNA. Amino acids corresponding to the codons are added to the growing polypeptide chain to form a protein. The code residing on the messenger RNA helps in the translation of proteins and the construction of the proteins. Here you can see the messenger RNA codons below and the anticodons on the upper surface brought by the amino acids attached to transfer RNA. When we sum up, the DNA gets transcribed into the messenger RNA. The message of the messenger RNA through the codons is translated into proteins. So what are the requirements of the translation procedure? One is the messenger RNA with the codons, the initiation factors, the elongation factors, the release factors, amino acyl transfer RNA synthetases, transfer RNA which is charged with the amino acids, the ribosomes, the ribosomal RNA and the ribosomal proteins. What is the mechanism of translation? We must know that the cellular factory responsible for synthesizing the protein is the ribosome. The ribosome consists of the structural RNAs and about 80 different proteins. In an inactive state, the ribosomal subunits exist as two parts. One is large subunit and a small subunit. Each subunit gets attached itself during the time of translation. When the subunit encounters the messenger RNA, the process of translation of messenger RNA to protein begins. Here you can see a larger subunit on the upper part and a smaller subunit. When the start of the translation occurs, these both subunits come together to form an entire ribosome. In prokaryotes, the ribosome is about 70S, consists of two subunits. The larger subunit is 50S and a smaller subunit is 30S. The ribosomes of the eukaryotes is 80S, which are larger, consisting of 60S subunits and 40S subunits. What is this S? S denotes the sedimentation coefficient, which is expressed as Swedberg unit. Here you can see the prokaryotic ribosome on the left side, which is 70S, and the largest subunit is 50S subunit and a smaller subunit is 30S subunit. The largest subunit consists of the 5S RNA, 23S RNA, and the smaller subunit in a prokaryotes consists of the 16S RNA. On the right side, you can see the eukaryotic ribosome, which is about 80S. The largest subunit is about 60S and a smaller subunit is about 40S. The 60S subunit consists of the 5S RNA, 5.8S RNA, and 28S RNA. The smaller subunit consists of the 18S RNA. Now, what is the reading frame? One of the alternative ways of dividing up a sequence of bases in DNA or RNA into codons is called the reading frame. Here you can see the reading frame 1, where it goes from 5' to 3'. Each codon, the three nitrogenous bases, codes for a specific amino acid. For example, AGU codes for serine, CUU codes for leucine, ACC codes for threonine, GCA codes for alanine, UUG codes for leucine, UGG codes for serine. In reading frame 2, one nitrogenous base is pushed and the frame is pushed towards the right side, and the reading frame shifting to the right side causes a change in the formation of the amino acids based on the coding of the codon. For example, GUC codes for valine, UUA codes for leucine, CCG codes for proline, CAU codes for histidine, UGU codes for cysteine. In reading frame 3, there is a shift of two nitrogenous bases where UCU codes for serine, UAC codes for tyrosine, CGC codes for arginine, AUU codes for isoleucine, and GUG codes for valine. What is an open reading frame? Any sequence of DNA or RNA beginning with the start codon and which can be translated into a protein is known as an open reading frame. Here you can see the open reading frame. You have a start codon and a stop codon, and the center is the coding gene region. In frame 1, you can see the different codons coding for different amino acids. The frame 2, there is a shift of one nitrogenous base towards the right side. In frame 3, there is a shift of two nitrogenous bases to the right side. What is a translational unit? A translational unit in messenger RNA is the sequence of RNA that is flanked by the start codon. The start codon is AUG, and the stop codon encodes for polypeptides. Here you can see the different types of codon from codon 1 to codon 7. Codon 1, which is the start codon, which codes for methionine. Codon 2 codes for arginine. Codon 3 codes for glycine. Codon 4 codes for serine. Codon 5 codes for proline. Codon 6 codes for threonine. And codon 7 is the stop codon. What is the untranslated region? Messenger RNA also has some additional sequences that are not translated and are referred to as untranslated regions. The untranslated regions are present at both sides, 5-N before the start codon and at 3-N after the stop codon. Here you can see the 5' to 3' messenger RNA with a 5' cap followed by a 5' untranslated region before the start codon. And there is a coding segment between the start codon and the stop codon. And after the stop codon, there is a 3' untranslated region followed by the poly A tail. The start codon AUG begins with the coding sequence and is led by a special transfer RNA that carries methionine. The initiated transfer RNA charged with methionine binds to the AUG start codon. Here you can see the two ribosomal subunits with the start codon AUG and the anticodon UAC attached to the transfer RNA charged with the methionine amino acid. The initiated transfer RNA in prokaryotes is N-formyl methionine, F-met, which is attached to the initiated transfer RNA, whereas in eukaryotes, unmodified methionine is used. Both initiated transfer RNA and the non-initiated transfer RNA are charged with methionine by the same methionine transfer RNA synthetase to give the methionine transfer RNA. Only the initiator methionine transfer RNA is modified by transformylase to give N-formyl methionine transfer RNA, which is, you can see in the image, the methionine transfer RNA is converted into N-formyl methionine transfer RNA by the presence of 10-formyl tetrahydrofolate. The 10-formyl tetrahydrofolate is converted into tetrahydrofolate, giving off the formyl group to the methionine transfer RNA. Now, what is SD sequence? The 5' end of the messenger RNA of the prokaryotes has a special sequence which precedes the initial AUG start codon of the messenger RNA. This ribosome binding site is called Shine-Dalgarno sequence or SD sequence. This sequence base pairs with the region of 16S ribosomal RNA of the small ribosomal subunit facilitating initiation. Here you can see before the start codon, the small ribosomal subunit being attached to the Shine-Dalgarno sequence. After that, the start codon is present to which the initiator transfer RNA bringing the formyl methionine is attached. The subunits of the ribosomes 30S and 50S are usually disassociated from each other when not involved in translation. Now let us discuss about the initiation phase of translation. In E. coli, the initiation begins with the formation of an initiation complex. The initiation complex consists of 30S subunits of the ribosome, a messenger RNA, the charged N-formyl methionine transfer RNA, 3 proteinaceous initiation factors, IF1, IF2, IF3, GTP for energy, guanine triphosphate, and magnesium. Here in this image you can see the requirements. The ribosome consisting of the large subunit and a small subunit, the GTP, the charged transfer RNA with the anticodon on one side and the formal methionine on the other side, the initiation factors IF1, IF2, IF3, the elongation factors EFTU and EFG, and the messenger RNA from 5' to 3' having the triplet codons. How it starts? The components that form the initiation complex interacts in a series of steps. IF3 first binds to the 30S subunit of the ribosome and allows the 30S subunit of the ribosome to bind to the messenger RNA. Another initiation protein, IF2, then enhances the binding of the charged formal methionine transfer RNA to the small subunit in response to the AUG triplet. Here in this image you can see the initiation complex consisting of the larger subunit of ribosome and a smaller subunit of the ribosome and having the messenger RNA and F-methionine charged transfer RNA being attached and the initiation factors 1, initiation factors 2 and initiation factors 3. This step sets the reading frame so that all subsequent groups of 3 ribonucleotides are translated accurately. The assembly of the ribosomal subunits, the messenger RNA and the transfer RNA represent the initiation complex. Once the initiation complex has been assembled, IF3 is released and allows the initiation complex to combine with the 50S ribosomal subunit to form the complete ribosome. The small ribosomal subunit binds to its recognition sequence on the messenger RNA, and the methionine charged transfer RNA binds the AUG initiation codon, completing the initiation complex. The large ribosomal subunit joins the initiation complex with methionine charged transfer RNA, now occupying the P site. You can see in the image how it is explained so clearly. In the process, a molecule of GTP is hydrolyzed, providing the required energy, and the initiation factors IF1 and IF2 and GDP are released. You can see in step 1, the charged methionine transfer RNA is brought to the smaller subunit of the ribosome, and in step 2, the attachment of the anticodon and the codon bonding is there, formation of the ribosome smaller subunit with the messenger RNA and a transfer RNA charged with methionine, and in step 3 the formation of the initiation complex occurs with the release of the initiation factors 1, 2 and 3 and the release of energy and the joining of the smaller subunit and larger subunit of the ribosome. Now next we will move on to the elongation phase. Elongation is the second phase of translation. Once both the subunits of the ribosomes are assembled with the messenger RNA, binding sites for 2 charged transfer RNA molecules are formed. The sites of the ribosome are referred to as amino acyl site, otherwise called the A site, the peptidyl site called the P site, and the exit site called the E site. The charge initiated transfer RNA binds to the P site. Here you can see in this image the E site, the P site and the A site. In addition, you can see the larger ribosomal subunit and smaller ribosomal subunit, and there is a messenger RNA binding groove in the smaller subunit. Now what is step 1 in the elongation? First is the hydrogen bond formation. In the prokaryotic translation, the second transfer RNA and the A site of the ribosome forms the hydrogen bonds between its anticodon and the second codon on the messenger RNA, which is step 1. Here you can see in this image the second transfer RNA charged and is brought and is fixed to the messenger RNA, and the hydrogen bonds are formed between the codon and the anticodon, between the transfer RNA and the messenger RNA. Now what are the requirements of step 1? It requires correct transfer RNA, another GTP and 2 proteins called elongation factors EFTS and EFTU. What is the step 2 in the elongation process? The peptide bond formation. Once the charged transfer RNA molecule is positioned at the A site, the enzyme peptidyl transferase catalyzes the formation of peptide bonds that link the 2 amino acids together. This is called step 2. A second transfer RNA with an anticodon complementary to the second codon binds to the A site of the ribosome, and the hydrogen bonds form between the base pairs, and the amino acid carried by the transfer RNA in the P site is transferred to the amino acid in the A site. And the ribosome acts as a ribozyme and catalyzes the formation of a peptide bond. The formation of a peptide bond is a condensation reaction. How this peptide bond is formed? Amino acid transfer RNA synthesis add amino acids in this according to the second genetic code. Amino acid covalently join to specific transfer RNA at terminal T-CCA sequence. Amino terminal end of the amino acid second transfer RNA attacks COO of the first amino acid transfer RNA to form a peptide bond. In step 2, break of the covalent bonds occurs. At the same time, the covalent bond between the amino acid and the transfer RNA occupying the P site is hydrolyzed, broken. The product of this reaction is a dipeptide which is attached to the 3' end of the transfer RNA still residing in the A site. For elongation to be repeated, the transfer RNA attached to the P site, which is now uncharged, is released from the large subunit. The uncharged transfer RNA moves through the E site on the ribosome. Here you can see the transfer RNA from the P site exits out through the E site, and a third charged transfer RNA is brought in. In step 3 elongation, the entire messenger RNA, transfer RNA, amino acid 1, amino acid 2 complex shifts in the direction of the P site by a distance of 3 nucleotides. This step requires several elongation factors and the energy derived from hydrolysis of GTP. This results in the third triplet of messenger RNA to accept another charged transfer RNA into A site. Here you can see the start codon and the transfer RNA with methionine, and the second amino acid being brought and attached to the second codon. And the polypeptide chain is formed in the third image with the transfer RNA exciting through the exit site and the third amino acid being brought to the third codon. The sequence of elongation is repeated over and over again in step 5, step 6, step 7. It goes on and on. An additional amino acid is added to the growing polypeptide chain. Each time messenger RNA advances through the ribosome. Here you can see how a polypeptide chain is formed above by the peptide bonds created and the new transfer RNA being brought in, and the ribosomes moves across the messenger RNA adding amino acids and the formation of a polypeptide chain. Once a polypeptide chain is assembled, it emerges out from the base of the large subunit. Now to describe the images of elongation, step 1, the second charge transfer RNA has entered the A site facilitated by EFTU. First elongation step commences. Step 2, the peptide bond is formed. Uncharged transfer RNA moves to the E site and subsequently out of the ribosome. The messenger RNA has been translocated 3 bases to the left, resulting in the transfer RNA bearing the dipeptide to shift into the P site. In step 3, the first elongation step is complete, facilitated by the EFG, and the third charged transfer RNA is ready to enter the A site. In step 4, the third charged transfer RNA has entered the A site, facilitated by EFTU. Second elongation step continues. Tripeptide formed. Second elongation step completed. Uncharged transfer RNA moves to E site. In step 6, polypeptide chain synthesized and exciting ribosome. Now going to the third phase, that is the termination phase of translation of protein synthesis. When one of the 3 stop codons appears in the A site of the ribosome, the termination occurs. Here you can see the appearance of the termination codon UAG, where the polypeptide chain synthesis stops or the translation stops. What is step 1 of termination? The terminal codon signals the action of GTP dependent release factor, which cleaves the polypeptide chain from the terminal transfer RNA, releasing it from the translational complex. Here you can see the appearance of the stop codon and the formation of the release factor. The stop codons are UAG, UAA or UGA. If any one of these appears, the release factor is formed and the transfer RNA is released out of the ribosome and the free polypeptide is also released from the ribosomes, and there will be separation of the two subunits of the ribosome and the release of the messenger RNA, and the release factor is also released from the complex. In step 2, the transfer RNA released from the ribosome dissociates into the two subunits. Here, to explain the images, you can see the termination procedure. In step 1, the transfer RNA and the polypeptide chain are released. In step 2, the GTP dependent termination factors are activated, components separated, and polypeptide folds into a protein molecule. Now, let us discuss about something that is concerned with the effect of antibiotics on the translation. Antibiotics are medicines that kill the bacteria or prevent their multiplication. The word antibiotic means against life. Antibiotics are of two types, bactericidal antibiotics and bacteriostatic antibiotics. Bactericidal antibiotics kill the bacteria, example penicillin. Bacteriostatic antibiotic contains the growth of bacteria, example erythromycin. Now, what is the mechanism of action of antibiotics in the prevention of protein synthesis? Many antibiotics do not allow the pathogenic bacteria to flourish in the animal host because they inhibit one or the other stage of bacterial protein synthesis. The antibiotic tetracycline inhibits the binding between the amino acid transfer RNA and the messenger RNA. Chloramphenicol inhibits the peptidyl transferase enzyme and formation of the peptide bonds. Here in this image you can see where the tetracyclines affect. They prevent the binding of transfer RNA to the A site on the messenger RNA and the formation of the ribosome complex. Whereas the chloramphenicol binds to the 50S subunit on the P site and inhibits the transpeptidation reaction. Now, streptomycin inhibits the initiation of translation and causes misreading. Streptomycin causes a structural change which interferes with the recognition site of the codon-anticodon interaction, resulting in misreading of the genetic message carried by the messenger RNA. The mechanism of inhibition is depicted in the image. You can see that the recognition site is distorted and streptomycin is bound to the 30S subunits. This inhibits the protein synthesis. Aminoglycosides like neomycin inhibits the interaction between the transfer RNA and the messenger RNA. Erythromycin interferes with the amino acid translocation, preventing the transfer of the transfer RNA bound at the A site to the P site of the ribosomal RNA complex. Today, we discussed in Molecular Genetics Part 8 about the translation of proteins and the effect of antibiotics on the translation. Thank you. Kindly subscribe, like, share and comment the channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.